All right, this is your photosynthesis lab pre-lab video. What you're going to be doing is determining factors that affect photosynthesis. Now, this is going to be a 2-day lab. The first, the lab itself is going to be conducted the same way both days. You'll just be testing different variables on day 1 than you will on day 2. So on day 1, everyone is doing the same lab. Okay, you're actually looking at 2 different environments. One is a light unboiled, a light boiled, and then also a dark unboiled. So, and by looking at those 3 samples, we're really getting a light versus dark, and then we're getting a boiled versus unboiled. Okay, so if I was to draw my 3 samples, okay, here I have my light, right, and then my light's unboiled, and then my dark, which is going to be covered up with a Dixie cup, is going to be the dark unboiled. So here's we're comparing our light and dark, and then we're going to have a third sample that's going to be boiled but light. So with these 2, we're testing light versus dark. With these 2, we're testing boiled versus unboiled. Okay, and as far as what's being boiled is our plant sample. Specifically, I'm going to grind up some spinach. Okay, so the second day you're going to be looking at whatever variable you want to. I just throw some examples out there. We have wavelength of light, how that affects photosynthesis, the concentration of carbon dioxide, pH, light intensity. You can choose whichever one you want to choose, but you're only choosing one. And actually, your lab report is going to have to address all 3. Okay, so you're gonna look at 3 different experiments, 2 on the first day and then 1 on the second day. As far as how you want to go about doing your day 2 lab, that's completely up to you. You guys are going to be responsible for coming up with a procedure. However, before you get too far in your procedure, I would definitely suggest you talk to me to make sure that what you are trying to do can be accomplished with the materials that we have. Okay? Now, how are we going to be determining what's our dependent factor here, or our dependent variable? And the truth is, how we're testing if photosynthesis is occurring or not is by the light reaction. Okay? Specifically, during photosystem 1, right, I am producing NADPH, right? NADP plus is being reduced to form NADPH. Okay? Well, what we're going to do in our samples is we're going to expose them to a pigment. Now, this pigment or this chemical is going to do the same thing that the NADP does. It's going to get reduced. Okay? And this chemical, and I'll go ahead and choose a blue color because the chemical, which is called DPIP, okay, you don't need to worry about the acronym, but DPIP is going to be reduced, right? And DPIP is blue, which means as it's in its oxidized state, it's blue, but as it gets reduced, it turns clear, right? So what we can show is if we can see the blue color fading to clear, we know that DPIP, which is acting like NADPH, right? Or NADP in this case, if DPIP is going from blue to clear, that is synonymous with NADP forming and P, and H plus forming NADPH. Okay, so the process of DPIP turning from blue to clear is the same thing as NADP turning into, or NADP plus, turning into NADPH. Okay, so this DPIP gets reduced, it turns clear, just like NADPH gets reduced and it will just be NADPH. The reason why we choose DPIP and not try to identify NADPH is because DPIP is a nice blue pigment that we can quantify using a spectrophotometer or using a calorimeter. Okay. Now, both of those, okay, spectrophotometer and calorimeter, they do the same thing. We are going to use a calorimeter. Okay. This is a calorimeter right here. Okay. Get rid of that line there. All right. So this is a calorimeter, or calorimeter. If you look, okay, you can see these little glass cuvettes. Okay, these cuvettes are going to contain your samples, right? The samples go into the calorimeter, and the calorimeter is going to be set at a specific wavelength of light, right? In this case, I want to say, it's kind of hard to read in that picture, but I want to say it's 635 nanometers. That's the one we're going to choose. We're going to choose 635 nanometers, all right? And as far as, you know, whether or not your calorimeter is set to that, you can click on the light blue arrows, or the light blue buttons, which have the white arrows on them as indicated with the orange arrow right here, right? This guy right here. You can click that, push on that until you go over to 635, okay? This center button right here, this calibrate button, we're going to use that to calibrate our calorimeter to make sure that we're getting accurate measurements to start. Okay, and I'll show you guys what your samples are going to look like and what's going to be in your different cuvettes to do your sample. Okay, so just another idea of how the calorimeter works. Okay, here is a side view and obviously a cross section of some sort, and here's my cuvette, which I've put in from top. I have a light source, and this light source is pushing out light at 395 nanometers wavelength. Okay. So as that light travels into your sample, okay, your sample, which is right here, okay, some of that light is going to be absorbed by the liquid that you have in there, by your sample, and some of it's going to be transmitted. Okay, so this detector that's inside the calorimeter is going to send the signal to your interface to tell you exactly how much of that particular wavelength of light was absorbed from your absorbed by your sample, right? Obviously, we should see a change in absorption as the change in color occurs as DPIP gets reduced. So as DPIP gets reduced, we should have a change in our absorption. Okay. So that's how it works. Okay, light goes in to the liquid. Liquid is going to either absorb some light, or it's going to absorb some light and reflect the rest of the light, or allow the light to be transmitted through it. And the detector is going to determine how much light is being transmitted, which is also going to give you the amount of light that's being absorbed, because whatever doesn't get transmitted was absorbed. Okay, so what's going to be in your samples? Okay, your samples are going to include, you're going to have four cuvettes. Okay, 3 of them are experimental, and 1 of them is a blank, and the blank is just for the calibration. Okay, so the blank, or what you'll indicate as a BL, okay, is going to have a milliliter of buffer, which is just to regulate pH. Okay, you're going to have 2 milliliters of distilled water, and you will have no DPIP. Okay, so that you'll have 3 milliliters of solution in there, and then right before you actually do the calibration, you're going to add 3 drops of unboiled chloroplast, and I'll have the buffer, the distilled water, and the DPIP, and the unboiled chloroplast available at your stations, okay, at your pods. So what I'd have you do is always add your chloroplast right, in this case, you add it just before you do the calibration. You don't let it just sit in there, okay? So you do your buffer, your distilled water, your DPIP, and there'd be no DPIP in this one. And then you add your 3 drops and you do the calibration. Now, just to kind of go back to explain what you would do, okay, you would put your cuvette in with the blank in it, close this top, okay, make sure this is set to 635 nanometers, okay, and then you push the calibration button, and you would just let it sit, okay, until it calibrates according to your interface, right? You look at your interface, and the absorbance will read 0.00. Okay, when that says, and that's done, then you're done with calibration. If you have to stop or you have to recalibrate it, then recalibrate it. But once you do the first calibration, you shouldn't have to do it again. All right. Now, one thing I want to mention with these cuvettes, right? They have a little cap. Make sure the cap goes on. Okay, you can actually see. I'll get the eraser. That's a little cap right there. Make sure your cap goes on the cuvette before you put it in the calorimeter. Also, when you're looking at this, okay, if you take a look at this cross-section diagram, it looks like there's little lines right here. Okay, it's because if I was to hold this up to you, obviously I can't because it's not three-dimensional, but in the z-axis on this side and on the other side, there are little lines, okay, that are going to indicate this side does not go where the light goes. Okay, the light should go on the clear side. So this side right here, all right, and this side right here should be clear, and that's the direction of the light. You don't want the light to hit these lines because then it'll just it'll mess up your results, not to mention disperse all the light. Okay, so that's how you would calibrate it. Then you're going to have an unboiled cuvette, a boiled. So unboiled cuvette is a milliliter buffer, milliliter still water, milliliter DPIP, and then right before we expose it to light, we'll add 3 drops of unboiled chloroplast. Okay, the dark cuvette is going to be the same thing as the unboiled cuvette, except we're going to cover it up with a Dixie cup so no light gets to it. Okay, and again, do the same thing, 1, 1, and 1, and then 3 drops of unboiled chloroplast right before you expose it to the light. And of course, that should be right before, or the light exposure should come right after you've covered it with the Dixie cup. Okay, and then your boiled cuvette is going to be 1 milliliter buffer, 1 milliliter distilled, 1 milliliter DPIP, and then you're going to add 3 drops of boiled chloroplast. And again, you will add those right before the light exposure. Okay. So what this should look like is I have my light source. Okay. So here's my light source coming out. Okay. It's going to be a heat lamp. Okay, with a 100-watt bulb. Right. And then you're going to have a large 2,000 milliliter beaker. That's going to have water in it. Okay? The reason why we have the water in it is because my cuvettes are going to be over here. I'm going to have 3 cuvettes. Obviously, they're not going to be stacked in front of each other. They're going to be next to each other where they're all exposed to the light. Okay? Obviously, remember, one of them is going to have a Dixie cup over it, which would be our dark one. But one thing you need to keep in mind is which one's which, because you can't write on the cuvettes and you can't write on the lids. So my suggestion would be to have a piece of tape on the bench and have it labeled with a D, a U, and a B so you know which one's which. Okay. Now you're going to be taking samples from this on a regular basis. Okay. You're going to be putting them in the calorimeter like every 5 minutes for 15 minutes. So you're going to want to make sure you put it back in the same spot. Okay. And the reason why we have the water right here is because the water is going to act as a heat sink. Okay, the water is going to absorb all the heat that's given off by the lamp, but it'll still allow the light to pass through. And of course, if we don't have the heat sink, then the samples are actually going to warm up, and we don't want that because we're not testing for temperature. We're testing for, in this case, dark versus light, and then boiled versus unboiled. Okay. So, as far as your day 2 stuff, it should come along the same lines as far as your basic setup. You're going to have a heat sink, you're going to have a lamp, you're going to have some samples, and then you'll have to recalibrate the next day. But it should follow the basic diagram like we have right here. We have a light, we have a heat sink, and we have our samples. Okay? And like I said, you're going to be taking samples 15 minutes, right? Every 5 minutes. So you should have a grand total of, in this case, we'll have 12 absorption measurements. Okay. Why are we having 12? Because we should have a time equals 0, time equals 5, time equals 10, and time equals 15. That's 4. You have 1, 2, 3 cuvettes. Okay, so every 5 minutes you're going to have to, and I'd probably stagger them, obviously. Probably go a minute and then do the next one, and then have another minute and do the next one. So you don't have to try to get all your things, because you can only put one cuvette in at a time, anyways. Okay, so you're going to want your cuvette 1. We'll go in, get a measurement, put it back. Then do cuvette 2, then do cuvette 3, and you're going to do 4 of them. 1 at 0, 1 at 5, 1 at 10, 1 at 20. Okay. If you guys have any questions about this particular portion of the lab, be sure to ask me either at lunch, or you can ask me during brunch, or right before class. But once class starts, we got to get going on it. Okay, I'll have everything ready for you, but I need you guys to have the basics down. All right. So again, of course, if you need to, you can always send me an email. All right. Bring closed-toed shoes.